here at the Plant and Animal Genome Conference in 2015. The question is, when is a worm not a worm? So Lex, I understand that the Western Corn Rootworm is not a worm, but actually a beetle larva. Can you help explain that?
Yeah, that's right. So it's one of those species, there's a lot of species out there that got names a long time ago, and phylogenetically the name is inaccurate. So it's a beetle species, but it has a common name that implies that it's a worm.
And it's a major problem, is that correct?
Yeah, it's the most dominant corn pest in North America.
It eats up corn roots, causes what? Corn to fall over from wind?
Yeah, that's right. So it chews off the roots as a larvae, and then as the plants mature, they don't have that robust root system and they can't get the water they need, and they also are really unstable in high wind.
I understand the genetics of the rootworm is really complicated.
Yeah. So rootworms, it's kind of interesting. They have a genome both in size and complexity that's an awful lot like corn. So their host species has a complex genome with lots of genes and lots of repeats. And the corn rootworm, the pest, has a big genome, almost the same size, with a lot of repeats and a lot of junk DNA, as they call it.
And so in your talk, you talked about some evidence that it was autosomal recessive, is that correct?
Yeah, so we're fairly confident now we've mapped resistance to a BT gene, and that we mapped it to a single chromosome, an autosomal chromosome, and it appears to behave in a nearly recessive manner. It's not perfectly recessive, but it's close.
And then you were zeroing in on that particular chromosome, that particular region, using AmpliSeq technology, or?
Yeah, so using genotyping-by-sequencing, we've been able to identify where that region is and to kind of whittle it down to a small portion of the genome. And now we can start to test hypotheses about what genes might actually be causing it, because we've reduced it to, I think it's something like a 50th of the total genome. So instead of working on this huge genome, now we're working on approximately 1/50th of its size, which makes it a smaller haystack to find our needle in.
Yeah, but that work is then still ongoing, is that correct?
That's right.
And then you mentioned before a little bit about the migration of this particular pest.
Yeah, so the Western Corn Rootworm is actually, it probably should more appropriately be called the Mexican Corn Rootworm. So corn evolved as maize in Mexico and was domesticated there and then spread across the Americas by Native Americans. And the Western Corn Rootworm is native to Mexico and most likely spread up through the desert Southwest and into the Americas. And as corn has moved across the globe, the corn rootworm, its major pest, has sort of chased after it.
And you're concerned as far as the particular BT resistance, as far as this corn rootworm goes, and as far as the goal of the research then, by finding mechanism and action, then what happens there?
Yeah, so we have two major goals. We want to understand how they've evolved, how the rootworms have evolved resistance to this BT toxin, because the BT toxin is one of the best control options we have. They're a very difficult pest to control. So we want to know how they've defeated this particular BT toxin, and that will help us design new products that have new modes of action so that those resistant bugs will now be susceptible. In addition, we want to ensure a long life and durability for this product. So we've caught the resistance issue pretty early. It's still very rare, and so we'd like to use genetic markers to be able to identify at-risk populations and try to stay out ahead of the problem. So rather than playing defense, we'd like to play offense and try to stop resistance before it spreads, because it's like many of these kind of epidemiological things, when it's small and localized, it's much easier to deal with than once it's widespread and common.
Yep. Certainly. Well, thank you very much for sharing your work today.
All right.
Very interesting.
Thanks, Dale.